Good afternoon, students. So, today we'll be dealing with new topics in our first chapter. So, before that, what all things we have discussed in our last class? It's about significant figures and the concentration, how to express concentration, that is normality, molality, percentage, etc. Then, types of titration, neutralization, precipitation, complexometry, etc. So today's topic, that is errors and types of errors, how to minimize errors, then accuracy and precision. So errors. What do you mean by errors? Errors means it's a deviation or it's a difference between true result and measured result. That is called errors. If errors in our analysis is very large means it results in many other serious consequences. For example, we can say that laboratory result. If any error came in that result mean the patient will undergo very expensive and dangerous medical treatment. So we should be very careful. So there are two types of errors, that is determinate errors and indeterminate errors. Determinate errors means it is caused by the our fault in any procedures or instruments, reagents, etc. So these can be corrected and can be avoided. So they are divided into many types. There is personal error, instrumental error, operational error, reagent error, and finally methodic error. Personal error. Personal error means we are responsible for the errors in the experiment. For example, if we are doing titration, if you didn't properly judge the color of the solution, it's our mistake. So these error may differ from person to person. So that is called personal error. Then instrumental error. Instrumental error means it is mainly due to instruments that we are using. For example, chemical balance or uncalibrated weights and graduated glass wires. Then operational error. Operational error means if we didn't follow proper procedure, the result may occur. Then reagent error. Reagent error means if we use expired reagents, or sometimes the reagents will react with our glasswares or containers. So this also may leads to errors. Or there are two types of reagents, that is laboratory reagents and analytical reagents. See, analytical reagents is more pure comparing with laboratory reagents. So if you're using analytical reagents, we can avoid error. And methodic error. Methodic error means based upon the method, what method we are choosing for our experiment. If there is any mistake in our method, error may occur. For example, in volumetric analysis, if the reaction didn't take place, if we didn't get any result, if we are not getting any color, if we titrate, it is mainly due to the occurrence of side reactions. Next one, indeterminate or random error. So these type of errors we cannot be corrected. So determinate error means we can correct it and we can find out where it become wrong and we can correct the error, but here we cannot do so. That is called a indeterminate error. We can call it as random error. It cannot be eliminated or it cannot be corrected. It is happening accidentally. Then minimization of errors. How to minimize errors? First one, calibration of apparatus. So it's very important to calibrate each and every apparatus periodically. So in our lab manual there is an experiment for the calibration. So at that time you will get good idea. Calibration of apparatus. Because without cleaning the apparatus, Sometimes we are doing experiment, it results in many problems, impurities will be there, or sometimes any wear and tear will be there in apparatus. So we should calibrate the apparatus periodically. Then analysis of the standard samples. So if we got any sample, we have to carry it out their analysis. Sometimes we go with synthesis for checking the samples, or it is purchased from National Bureau of Standards. Then running a blank determination. Blank determination means sample is omitted. That means there will be no sample. Other than sample, reagents, solvents, it will be there. So when we are running a blank determination, we could understand whether there are any contaminants is present in our reagents or in our vessels, etc. In the case of titration, we could make an correction. That means if any excess reagent is present in our solution, we could find it out. Then independent analysis. Independent analysis means we are doing two times. If I have done one method, that is, I first of all I used volumetric method. Then I want to check it whether it is right or not. So I went for another method, precipitation of gravimetric method, like that. For example, you can see that I have to check the strength of hydrochloric acid. So first of all I go with titration. Then I went to gravimetric method. Then I'll compare the two results. So if the results are accurate, then my method is good. If any error came means my method will be wrong. That is independent analysis. Next, standard addition. Standard addition means we are adding a constant or a substance to a sample. Then totally we'll analyze that particular substance. And as a next step, what we'll do, without adding constant, we'll analyze that sample and we used to compare that results. That is called a standard addition. Then internal standard. Internal standard means it is mainly for chromatographic method or spectroscopic methods. Chromatographic method means paper chromatography. There are many methods. Spectroscopic method means IR method, UV method, etc. Actually here we have lot of samples and we'll check the concentration of each and every sample, and we used to plot the graph, so we'll get a straight line. So another concentration, that means we don't know which concentration, that is unknown concentration. If unknown concentration is there means we used to plot by extrapolation method. The graphical method will change. That is internal standard. Next, amplification method. Amplification method means to check the analysis of the very minute substance. That is called the amplification method. Then isotopic dilution. Isotopic dilution means here we are adding radioactive isotope to a sample. Then element. Then it is isolated into pure form. And finally we will measure that radioactivity. Then we add another element with that and we will compare the concentration of the element in the sample. Then accuracy. Accuracy means it is the correctness of measurement between the true value of accepted value. So accuracy can be expressed in two modes, that is absolute error and relative error. So there is a formula, E is equal to xi minus xt, and the relative error means xi minus xt divided by xi into 100%. XT means true value. XI means mean value. Then precision. What do you mean by precision? Precision means how close they can be each other. That is precision. For example, we are measuring a substance using a weighing balance 3 or 5 times. So I'm getting 3.2 kg each time. Then my measurement is precise. Actually, precision is based on the measuring tool. Accuracy means it is based on the measuring person. So precision means it is independent of accuracy. Sometimes we can be precise but inaccurate. Sometimes it will be accurate but it may be imprecise. So you can see it from our figure. See, this is having good accuracy and precision because the values are very close to each other. And here the precision but without accuracy. See, it's not accurate but it is very precise. Then accuracy without precision. See, so these values are near but it is not precise because it is not close to that much close to each other. Here no accuracy and no precision. Last figure. So this is accuracy and precision. And that's for today. Thank you.